Today's lab is about photosynthesis and chlorophyll, especially we're interested today in the effect of different wavelengths of light on chlorophyll. Some of the wavelengths of light are better at doing photosynthesis, and some are worse. We're actually going to do an experiment to find out which are better and which are worse. Let's start out by defining photosynthesis. Basically, photosynthesis is when photosynthetic organisms, like most plants, take light energy, and they use that light energy to manufacture their food. Their food is in the form of glucose or some other carbohydrate. And the raw material they use to make their food is carbon dioxide, gaseous carbon dioxide. We're going to go into detail a little bit about the anatomy of a leaf and how it takes in carbon dioxide. Okay. But first, we're going to talk a little bit more about photosynthesis. The primary photosynthetic pigment is chlorophyll. Chlorophyll is green in color. However, there are other photosynthetic pigments other than chlorophyll. And a photosynthetic pigment is a pigment that's got a particular color. And each pigment specializes in absorbing certain wavelengths of light. Wavelengths of light it does not absorb, it will reflect. If you have more absorbance at a particular wavelength, that wavelength is more suitable for doing photosynthesis. If it reflects or transmits through, it's not nearly as useful for photosynthesis. And let's think a little bit about pigments. So this notebook is covered with red pigment, and because it's red because it reflects red wavelengths of light and absorbs other wavelengths of light, other colors. White light, like we have from our lamps or from the sun, is a mixture of all different wavelengths of light. That is, it's a mixture of all different colors of light all together. When the white light strikes the pigment, for this particular notebook, red wavelengths of light bounce off and hit our eyes. That's why we see it as red. Other colors of light are absorbed. So from this, we can predict that since chlorophyll is a green pigment, it probably does not do much absorption of green wavelengths of light. It reflects them, and therefore green light's not very good for photosynthesis. But we'll find that out experimentally in just a minute. Okay, the first thing we're looking at is leaf anatomy. So let's take a look at the video monitor here. We have a cross-section of a typical plant leaf, and the layers you need to know. We've got the upper epidermis and the lower epidermis. The upper epidermis and the lower epidermis' primary job is to protect the plant. And next we have the cuticle. It's not very easily visible on the monitor, but a cuticle is a waxy layer on the surface of the leaf. The primary purpose of the cuticle is to prevent evaporation of water, so it's to prevent the leaf from drying up. On this particular slide, the red color represents chloroplasts because our chloroplasts have been stained red. So this red layer here is the palisade mesophyll. The primary purpose of our palisade mesophyll is to do photosynthesis. That's why it's so heavily pink in color. This layer right here is the spongy mesophyll. Spongy mesophyll has got a lot of air spaces. It's very loosely packed. The spongy mesophyll's primary purpose is to absorb carbon dioxide. That's why it has so many air spaces. Carbon dioxide can circulate through, get dissolved by absorbed by the different cells. And let's see, next we have vascular bundles, and I'm going to find you a vascular bundle. Okay, good. So this is a vascular bundle on a leaf right here. You can see basically it acts like the circulatory system of the leaf. It transmits water, dissolved minerals, dissolved food, dissolved carbon dioxide, and things throughout the leaf. And the next thing we're going to look at is a stomata. I'll find you a stomata. Okay, good. Here's a stomata. It's an opening on the underside of the leaf, and its purpose is to allow carbon dioxide to diffuse into the spongy mesophyll and therefore get absorbed. The stomata has two different cells associated with it. It's got a guard cell on either side. Guard cells will open the stomata and they'll close the stomata. They open the stomata to let in CO2. They close them to prevent too much carbon dioxide being absorbed. Okay. Our next experiment was to make a slide of another view of the stomata. We did it by painting some clear fingernail polish on the underside of a leaf. Then we peeled off the fingernail polish and made a wet mount slide of it to see an impression of what the stomata look like. Put one on so we can see what it looks like. Zoom in. Okay. Good. See, these oval-shaped structures are the stomata. You see we have a guard cell here and a guard cell here. Those two guard cells will open and they will close the stomata. So you should be able to pick out whether the stomata are open or closed. Looks like our stomata are closed. See, it's only a very slit for the opening. So when they close, they're trying to save water. When they open, they're trying to let in carbon dioxide. Okay. So our next experiment was paper chromatography. And let's walk over here and let's see what paper chromatography is all about. Okay, paper chromatography is a way of separating out different pigments, and we separate them out actually by molecular weight. So we got some strips of paper chromatography paper, and we rubbed spinach juice in a band about 1 inch from the bottom of the strip. Our spinach juice, we just smashed it in there, so we got a nice green line on our strip. And then we stuck our paper inside a jar of solvent, like so. The idea is that this solvent is going to wet the paper strip, it's going to wick up it, and it's going to dissolve all the spinach juice that is mashed onto the paper. And there are actually multiple photosynthetic pigments on the paper. There's actually at least 4 photosynthetic pigments in our spinach juice. There's chlorophyll A, chlorophyll B, carotenoid, and xanthophyll. And as it dissolves the photosynthetic pigments, it carries them upward with it as it continues to wick up the strip. And those pigments will separate out into separate bands depending on their molecular weights. The heavier pigments will stay toward the bottom, the lighter pigments will travel to the top. So the 2 yellow bands are the lighter ones, and they'll be at the top. I believe it's carotenoid is the lightest band, so it's the very top yellow band. And the heaviest band is one of the chlorophylls, and it's the very bottom. So that's the heaviest band of photosynthetic pigments. And we have a lighter one here. We have a light yellow one here, and we have the very lightest photosynthetic pigment at the top. So one here, one here, one here, and one here. They haven't separated out completely. The bottom 3 haven't. Okay, so that is paper chromatography. We're separating out our 4 photosynthetic pigments: chlorophyll A, chlorophyll B, xanthophyll, and carotenoid. Let's walk over here and we'll take a look at our spectrophotometer. Okay, our spectrophotometer here is an instrument, and it measures the absorbance of our specimen at different wavelengths of light. So what we're interested in is the absorbance of chlorophyll at different wavelengths of light. Remember, if you have more absorbance, that particular wavelength of light is better for photosynthesis. If you have less absorbance, it's not as good. So we actually wanted to find which wavelengths of light are better than others. So we actually had a problem we had to solve. This is chlorophyll. This is dissolved in petroleum ether. It's in a glass tube. We don't want to know the absorbance of the petroleum ether in the glass tube. We only care about the absorbance of the chlorophyll. So we solved that by using a blank with petroleum ether and glass tube only. So we could zero it out, so that is subtract the absorbance of those two, of the petroleum ether in the glass before we took our readings. So all we did was we cleaned off our tube, we inserted it in here, and we changed the wavelength to wavelength of light. First one was 400. So 400 wavelength is purple light. So I'm going to turn this dial and zero it out. So now I'm subtracting the absorbance of petroleum ether in the glass. So it's at 0. Pull it out. Now I'm going to clean off my chlorophyll tube. When we put it in, it's going to tell us absorbance of chlorophyll only and not petroleum ether of the glass. So our absorbance with the purple light is about 1.32%. So you would need to work through and do all the rest of our wavelengths to find out, relatively speaking, which ones are better, which ones are worse. As you would predict, the green green wavelengths of light, that is about wavelength 480 to 520, 540, is not very good for absorbing light. And we could have predicted that because chlorophyll is green, so it reflects green light rather than absorbing it. Okay. Let's see. Our next demo was a demo of chlorophyll fluorescence. Let's walk over here. Okay, we've got some extracted chlorophyll. So this chlorophyll is not a part of a living leaf anymore. You can see it is, of course, green in color. But it's been separated by a leaf, so it cannot do photosynthesis. However, it's still capable of absorbing some light. Now, obviously, when a leaf absorbs light, what it wants to do is photosynthesis. However, sometimes it's prevented from doing photosynthesis. For example, our chlorophyll here is taken out of a leaf. It can't do photosynthesis anymore. But it's still capable of absorbing light. So when it absorbs light, it's got to do something with that energy it's absorbed. And it's going to do one of two things. If it can't do photosynthesis, it will release that energy in the form of waste heat to the atmosphere, or release it in the form of waste heat and fluorescence of light. So I'll shine the UV light on here, and we can see that our chlorophyll is actually glowing red in color from the UV light. Because chlorophyll atoms are getting excited by the UV light, and they can't do photosynthesis with the light, and so they are going to release that energy in the form of waste heat and also waste light. Okay, so that's all the experiments we did today in the photosynthesis lab.